Okay, so we talked about a lot of things yesterday. We talked about the entire muscle and how it's divided into fascicles, and each fascicle is composed of fibers, and how the connective tissues of the muscle are continuous with the connective tissue of the nerve, or excuse me, of the tendon, and parallel to the ones of a nerve. We also got down into an individual muscle fiber, and we saw that it's made of bundles of proteins. Those proteins are called myofilaments, and the bundles are called myofibrils. And we looked at the structure of a sarcomere and how the sliding of the filament shortens with force. And we had already met the membranous components of a cell, which are the sarcolemma, which is the plasma membrane, the transverse or T-tubules, and the sarcoplasmic reticulum. And we're going to come back and hit that again when we look at what the heck are they doing there, okay? So, but we're going to start back with the nerve that's stimulating that muscle. And you remember that every muscle gets its own nerve, and every nerve branches within the muscle. You get a branch to each fascicle, and within the fascicle you get individual nerve endings for each muscle fiber. So we're going to look at that from the standpoint of a single unit. We call it a motor unit. Anytime we're talking motor, what we're talking about is the branch of the nervous system that sends stimulation out to make muscles contract or glands secrete. So in a motor unit, the cell that's in charge of everything is the motor neuron. Those are the nerve cells. And when you look at nerve cells, you find that the ones that stimulate muscles are sort of a star-shaped neuron. And they're going to be in either the brain or the spinal cord. It depends on where they're going. So if they're going out to the body, then they tend to come out the spinal cord. But if they're going like to the muscles of your face or to blink your eyes or whatever, then those are going to come off the brain. So we'll worry about the classification of the neurons later. But the nerve fiber that comes from that neuron is called an axon. The axon is the nerve fiber. And each nerve fiber is the axon of an individual neuron, and then these are going to bundle up into nerves. So a nerve is a bundle of axons. All right, when it gets into the muscle and into the fascicle, that axon can actually branch. So we're already in the muscle when it does that. And each branch can stimulate a different muscle fiber. So this particular motor neuron, poorly drawn, is stimulating 3 muscle fibers. So when that neuron gets an impulse and that impulse travels down the axon, when it gets to the branch point, it goes through all 3 branches at the same time and stimulates all 3 muscle fibers at the same time. Okay? So a motor unit is one motor neuron plus all the muscle fibers it connects to, however many that may be. Does everybody see the point of that? Did you learn that in lab today, this morning? Motor unit? They didn't cover motor units? Okay. Okay, the number of muscle fibers that is controlled by a single motor neuron is different for different needs. So, for example, if you were talking about the gluteus maximus muscle, it's a very large, powerful muscle. Would it do you much good to have one motor unit that controlled only 5 muscle fibers out of that huge, massive muscle? Is that going to have much of an effect? Probably not. Okay, so it's probably more on the order of 1,000 muscle fibers in the gluteus maximus. Honestly, I've never looked it up, but probably in that range. On the other hand, these little muscles that make your eyes twitch when you get stressed too much, like taking summer school, those are little tiny muscles. So does it make sense to you that they would have smaller motor units? That is, motor units with fewer muscle cells in them. So the number of muscle fibers per motor unit is not fixed. It varies and it depends on the muscle and the needs of that muscle. Does that make sense to everybody? All right, so what I want you to do in your groups is explain why you would need different numbers of muscle fibers in a motor unit in the same size muscle that moves your thumb as the one that moves your great toe. Okay, so I've got parallel muscles that move my thumb. I can do some of the same movements with my great toe. I will not be demonstrating that. Use your imagination, but tell me why you wouldn't have the same size motor units in muscles of the thumb compared to muscles of the great toe. The muscles are the same size, so why would you need different numbers of motor units? Go. Great toe, big toe. So if you're trying to follow along with the PDF, I think we're on page 6. All right, so what do you know about the difference in humans between the thumb and the great toe? You use your upper limb, the manus, more than you use the pedis, right? Okay, so what difference does that make? You need more control over thumb than you need over toe. Okay? So why does that translate into I need more or fewer? Which one? More motor units in the thumb or fewer motor units? More motor units in the thumb, what does that give me? More control in what way? I mean, if I have more muscles, that's a different question. Do I have more muscles that control my thumb than I have controlling my great toe? You know, never really looked, but I'm just guessing yes. Why? What can I do with my thumb? It's opposable. Whereas in humans, the great toes, and I keep saying in humans because our primate cousins, the great apes, do have opposable toes. They have very good grasping. Not so good at walking long distances, but better at grasping. So, and we'll come back to that. All right, so that's a different question, though. Do I have the same numbers of muscles? No. Do I have the same movements? No. But if I took the corresponding muscle that does, say, flexion of the thumb, because I can flex my great toe. If I look just at that muscle, why would I have different numbers of motor units in the muscle of the thumb than I would have in muscles of the toe? Well, first of all, would I have more or less? What does more motor units tell you? Okay, so thinking of an individual muscle, if it's divided into 10 motor units, then I can have 10 different strengths of contractions. Everybody see the point I'm making now? So I could contract them all and get 100% contraction. I could contract just one of them and have a 10% contraction or anything in between, right? What if I only had 5 motor units? Would I have 10 different strengths of contraction? No, I'd have only 5, right? I can contract 1, 2, 3, 4, or all 5 of them, okay? So if I had the same number of muscle fibers in each muscle, the one of the thumb and the one of the great toe, if I had the same number of muscle fibers in each, but I had fewer motor units in my toe muscle, then I would have greater strength of intermediate contraction, but I wouldn't have as much of a degree of selection. Does that make sense or not? So what can I do with the musculature of my hand in terms of different degrees of strength of contraction? If I had to grasp something and hold a weight, I would want a really powerful contraction. So I would contract all the motor units. Okay, what if I'm holding an egg in my hand and I really don't want to mess? Now I just want a very light contraction. So I have different degrees of strength of contraction by having smaller, therefore more motor units. The more motor units a muscle has, the more control it has on how strongly it's going to contract. Okay? That's the big picture idea. All right? So as we'll see next week, when we map that to the brain, if I have more motor neurons controlling my hand than I have controlling my foot, then I need more brain to control my hand than I need to control my foot. Does that make sense to everybody? So the more control, the more precise control you have over a muscle, the more motor units you need to supply that muscle with. The more motor units, the more motor neurons it takes. The more motor neurons it takes, the more real estate in your brain you have to devote to that. So hold that thought till next week. We'll look at a map of the brain and we'll see what difference that makes. Okay, any questions about the idea of motor units? Okay, so please don't think of all the muscle fibers as being in the same space either. So if I have 10 motor units, then and each one's controlling 1,000 muscle fibers, the 1,000 muscle fibers of that motor unit are going to be scattered throughout the muscle so that you won't get contraction on one side of the muscle with one motor unit and a totally different part of the muscle with a different motor unit. They're going to be scattered, dispersed. Okay, so now we're going to follow our axon. Remember, that's the nerve fiber. We're going to follow it to its end. And when we do, remember that we're going to see it's going to branch. So as it comes into the muscle, it branches, but those branches are going to disperse throughout the muscle to hit muscle fibers scattered throughout the muscle. But if I follow just one of those close up, what I'm going to see at each one is that it branches again. And it branches over the surface of that individual muscle fiber that it's supplying. So here's the muscle fiber. And I have these branched endings of my nerve fiber, and each one where it ends in a little swelling. How much surface area do I have because I branched that nerve ending? In this picture, can you see I have 4 times as much surface area as I would have if I didn't have 4 branches? Okay, so what you have is a little cluster of nerve endings at one little spot in the middle of the muscle fiber. And that little spot is called the motor end plate. The motor end plate is the region, of the muscle fiber where the nerve connects to it, the nerve ending hits. When I look at it at just one of those little nerve endings close up, that little swelling that I see is called the synaptic knob. And you need to know what a synapse is. A synapse is a connection between a neuron and the cell that it's stimulating. So if I draw the motor end plate in the same level of detail that I'm drawing the nerve ending, what I'm going to see is even more remarkable. Here is the sarcolemma. What was the sarcolemma? That's the membrane of the muscle cell. And when it approaches the synaptic knob, it invaginates. What does that mean? It folds inward. And when it comes to the business end of the synaptic knob, it folds again. Look at these folds. Why do I have folds here? That's giving me so much surface area. It's just unbelievable. Okay? So those folds are below the neuron, so we call them subneural folds. Are they part of the muscle or the nerve? They're part of the muscle. So they are the functional portion of the motor end plate. Are you with me so far? Okay, so I had branches, right? Each branch ended in a knob. So I already had multiple knobs associated with one muscle fiber at the motor end plate. And then deep to each synaptic knob are these subneural folds, which is the membrane of the muscle cell. And finally, I have, notice please, a gap between, that's not showing up very well, between the nerve cell and the muscle cell. Does everybody see that gap? What do you want to call that? That's the synaptic cleft. The cells literally do not touch each other. So that cleft is a gap between the membrane of the neuron and the membrane of the muscle fiber. Questions about that? All right, so as we get into this, we're going to see where the action is. If we look at the synaptic knob, we're going to see that it has 2 or 3 different players that we need to be aware of. So if I draw again a synaptic knob, then what I'm going to find inside of it are vesicles. Do y'all remember what vesicles are? They're little bubbles of membrane that are full of something. These are called synaptic vesicles. And what they're filled with is the chemical messenger, which the class of chemical messenger is called the neurotransmitter. Have you heard that term before? The neurotransmitter is the chemical that's secreted by the neuron to stimulate, in this case, a muscle cell. And in the case of neuromuscular junctions, the synapses between a neuron and a skeletal muscle, I'm talking just about skeletal muscle, the chemical neurotransmitter is always acetylcholine, and we abbreviate acetylcholine, A for acetyl, and CH for choline. Can you tell me the 2 parts of the molecule of acetylcholine? This is a trick question. There's a choline part, and there's a little acetyl group attached to it. Okay, so what are my vesicles full of? The neurotransmitter, in this case specifically acetylcholine. The second thing I need to have in this cell is some kind of trigger that tells my cell when it's time to secrete its neurotransmitter. And the trigger for this process is going to be calcium. So at this point, we've seen calcium in so many different arenas that it's time to make just a general statement about calcium. When you let calcium into a cell, something's going to move. Okay? When calcium gets into a cell, some movement is going to occur. In a muscle cell, it causes contraction. In a secretory process like this, it causes exocytosis. So whether it's a gland cell or a nerve ending, when you let calcium into the cell, those synaptic vesicles go on the move. If I stimulate it a little bit, just a few of the synaptic vesicles will release their contents. If I re-stimulate it again and again, then I'm going to dump more and more and more of the synaptic contents. Can everybody picture that okay? A little bit of calcium, a little bit of secretion, lots of calcium, lots of secretion. Are you with me on that? We're going to learn the beginning of this story next week. How does the neuron do what it does? Right now, we're just focusing on the ends of things, and that makes it a little complicated to pick up the story in a place that makes sense. But where we are now is that at the synaptic knob, there are voltage-gated calcium channels. So at my nerve ending, here's my synaptic knob. Right there at the nerve ending, I have, in this area, some voltage-regulated calcium channels. Now, we haven't met these guys yet. So let's take that term apart. If it's a calcium channel, what do you know about it? It lets calcium either in or out of the cell. We don't know which direction yet. If I tell you it's voltage regulated, anything that's a regulated channel is a gate. Okay, so this is a gated channel. That means I have to open the gates. That's how I regulate it. What opens the gates in a voltage regulated channel? This is a trick question. Voltage. So where do I get voltage from? Well, this is a nerve cell, right? So don't I have electrical impulses passing down the cell? And guess what their unit of measure is? That would be volts. So when the voltage of the electrical impulse coming down the nerve fiber hits these gates, it causes them to open. So what then is going to happen next? Calcium's going to go one way or another, and let's figure out which way it's going to go. Calcium, when it gets into cells, causes things to move. If calcium were already in the cell, stuff would already be moving. So where does most of the calcium stay when a cell is not being stimulated? Outside the cell, in the extracellular fluid. So when I open these gates, calcium enters the cell. And what is that calcium going to do when it comes in? It triggers exocytosis of some, but not all, of the synaptic vesicles. So here's my acetylcholine. It's been sitting in those vesicles. Once I trigger its release, out it goes. What does it go out into? What do we call that little space? Synaptic cleft. So it goes out into the synaptic cleft. All right? The rest of the neuron story, we're going to try to save for later. I might have to bring up a few more details. It would be nice if you remember this part next week. So let's just review it. We had a motor neuron. It could be in the spinal cord or the brain, depending on where it's going. The nerve fibers, the axons, are out to the muscles. They branch within a muscle and go out to their muscle fibers that are part of that motor unit. They're all scattered. When it gets to an individual muscle fiber, it branches, I don't know how many times, 3 or 4, and each branch ends in a synaptic knob. Okay, so when the nerve impulse reaches the synaptic knob, what happens? Okay, what did I need before I could do the calcium doing that? I had to get the voltage there, right? So once the nerve impulse gets there, the voltage is there. What does the voltage trigger? The opening of the gates. So first I have to open the gates, then the calcium can flood into the cell, triggering exocytosis of synaptic vesicles. That releases the acetylcholine into the synaptic cleft. Are you with me on that? You should probably think of these voltage gates as being like on a timer. They're probably not really, but have you ever seen a gate that opened and then it would close automatically after a certain period of time? What really is probably happening is that the ions are moving around and the voltage disappears, and when the voltage change is gone, the gates close automatically. But just think of those gates as closing by themselves, just based on changes in the electrical charge. Are you with me on that? So once the calcium gates are closed, if I wanted to secrete more acetylcholine, what would I need to do? I need to send another impulse. So if I want to stimulate a muscle to contract with great force, then my nerves are repeatedly sending one impulse after another. Does that make sense? Can you picture that? So to strongly stimulate a muscle, I'm sending boom, 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 one nerve impulse after another. And as each nerve impulse reaches the synaptic knob, calcium gates open, little trickle of calcium in, the calcium gates close. What makes them reopen again? Next nerve impulse hits, they reopen and then close. Okay so far? All right, so just to make sure we have the complete story here, can I leave that calcium hanging around? What would happen if I did? As long as it sits in that cell, even though the gates have closed, as long as the calcium sits there, exocytosis continues. So what I'm going to have is the opposite of a calcium gate. What is the opposite of a calcium gate? Y'all need to know the opposite of gate. Opposite of gate is the thing that carries it the opposite direction. A gate is part of a channel. It moves by diffusion. How do I get stuff to go against diffusion? I have to use active transport. What do we call an active transporter? It's called a pump. Okay? So I have calcium voltage gated channels to let the calcium in, and then I have calcium pumps to push the calcium back out. Okay? And in general, where there are gates, there must also be pumps so that you can get rid of the stuff that you just let in. Does that make sense as far as that goes? All pumps require ATP. So for every nerve impulse that's generated, that nerve ending needs some ATP. What is it used to generate ATP? How do you generate ATP in a cell? Mitochondria. So do I have mitochondria in my synaptic knob? You betcha. I do. And that means I need a good blood supply too, right? To bring in the oxygen. Okay, so now you have a fairly complete picture of what's happening in a synaptic knob. Let's go back through it again. So when the nerve impulse hit, that changed the voltage, that opened the voltage-regulated gated channels, that opens the gates, calcium floods into the synaptic knob. That triggers exocytosis of some, but not all, of the synaptic vesicles. Okay, maybe just a few. There may be 1,000 of them packed in there, and we're just going to open a few. Okay, gates close automatically as the voltage passes. Pumps pump the calcium back out, exocytosis stops. Nerve ending is like, whew, done. I'm taking a break. Okay? So, but that's the story so far. And if I want to release more acetylcholine, I re-stimulate it, and I just do the same thing over and over again. Are you with me on that? Very good. So we're going to follow what the acetylcholine is doing. As soon as I get my control back. All right, so we're going to turn our attention to the motor end plate. What did it look like? Yep. So around the synapse, I had lots of folds. What did I call them? Your book just says numerous folds, but there's a name for them. They're called subneural folds because they're up under the nerve ending. And why do we have those folds there? Increases the surface area. So boy, is that surface area up to something. That thing is just packed. And then, remember, this is just a cross-sectional view. So thinking three-dimensionally around that entire synaptic knob, how folded is that membrane? How much membrane do you have? So what we find packed into that membrane is acetylcholine receptors. That should make sense, right? So when I had my nerve ending and I secreted that acetylcholine diffuses fairly rapidly down into my subneural folds. And when it gets down there, it's going to bind to its receptors. And that's going to trigger muscle contraction. Are you with me on that big picture? Acetylcholine gets into those folds, bind to its receptors. Somehow, we'll figure out how in just a few minutes, somehow that triggers contraction. So when I want the muscle to relax, what do I need to do? Get rid of the acetylcholine. Just like in the nerve ending, I had to get rid of the calcium. I was using calcium as a messenger there. You've got to remove that stuff. Likewise, acetylcholine is my messenger here. I have to have a way to get rid of it. It's going to be gotten rid of fairly quickly so that each stimulation lasts about a millisecond and a half. Okay, how long is a millisecond? It's 1,000th of a second. Okay, so each acetylcholine release is very, very short-lived. Okay, so let's take a look at our acetylcholine receptors, knowing that that's where the action is. We'll come back to that. Why is this not a great picture? How many folds did they give us? Not very many. I'll tell Valerie next time I talk to her. All right, so here we are in one highly stylized fold. And you can see that it has lots of these little guys here. What are these things? Those are channels, clearly. But they're labeled acetylcholine receptor. What's up with that? This particular protein is a hybrid. It has two different functions. It is both receptor and channel. So what do you suppose that channel does when acetylcholine binds to its receptor? That's what makes it open the channel. We call this a direct neurotransmitter function, direct neurotransmitter, because the neurotransmitter actually binds to the channel and makes it open. But that means that each channel protein is part channel and part receptor. So you can see, let's see if we have a close-up of that. You can see when you look at that picture that there are acetylcholine receptors. No. That are on the sides of the receptor protein. No, we're going too far. All right, so in this lovely diagram, we can see a lot of what we've already talked about. So what do you see here? It's, in case you can't read from where you are, voltage-gated calcium channels. So what happened? We got the nerve impulse, the change in voltage open these channels. Calcium rushes into the cell. What does calcium come in do to the cell? Triggers exocytosis, so the calcium causes the vesicle with ACH in it to bind to the membrane and release the acetylcholine. What does the acetylcholine do? Binds to its receptors. What happens when acetylcholine binds to its receptors? That opens this channel. And that's as far as we've got with that part of the story. Meanwhile, back up here, what's happening? Calcium's getting pumped back out. That stops the release of any more neurotransmitter. How long does all that take? About a millisecond. Everything's going to be about a millisecond, okay? All right. So now we get to turn our attention here to these channels. And what those channels are going to do when they open is they're going to allow the flow of both sodium and potassium. These are sodium-potassium channels. Which way are sodium and potassium going to move? Opposite each other. Does anybody have a clue which way they move? In a cell, typically. Did we meet something we call the sodium-potassium pump? What did it do? Pump sodium and potassium. Okay, that's part one. Which way did it pump each one? Y'all cannot forget this again. Okay, just like you cannot forget which way pH changes when you add acid, which is what, by the way? pH goes down when you add acid. Do not forget that. I've already had problems in AP2 this semester because of people missing that. Okay, y'all aren't going to do that. It would reflect poorly on my instructional skills. Okay, so likewise, you're not going to forget this. The sodium-potassium pump pumps 3 sodiums out and what? 2 potassiums in. If you're an artist, make a painting of this or a drawing. If you do needlework, stitch it. Okay, make a lovely diagram of this. Some way or another, do not ever forget it again. The sodium-potassium pump is working in a cell pretty much tonically, which means all the time. And it's sitting there just doing its little thing. And you don't have just a few of these, you have a lot of them. So you have a lot of these pumps, all of them doing the same thing. They eject 3 sodiums, they bring in 2 potassiums. They use an ATP every time they do. And the net result of that is that you have a lot of sodium on the outside of the cell because you're continually pumping it out. And then what else do you have? Lots of potassium on the inside of the cell because you're constantly pumping it in. Okay? So fix that in your minds. So if I drew a cell and asked you, where is there a high concentration of sodium? Outside, thank you. So sodium concentration very high on the outside. What about sodium on the inside? Very low. So we'll just draw that really teeny tiny. And then what about the potassium? Very high on the inside, very low on the outside. And why is that? Those pumps. Since you were a fertilized egg, they've been going nonstop your whole life. Okay? So consequently, what is the concentration gradient for sodium? Which way would sodium move if I open a channel? It wants to diffuse in because I've been pumping it out. And if the same channel let potassium move, which way would the potassium move? It tends to move out. Is everybody okay with those diffusion gradients? Questions at this point? So what made those diffusion gradients exist? It was the pump. Okay, so we blame it all on the pump. Got it? And how many in which direction for that pump? 3 sodiums out, 2 potassiums in. And it's not an equal amount, right? So because of that and a variety of other reasons, the sodium wants to move in more than the potassium wants to move out. Can everybody see that that would be the case? Even though we don't have a complete picture of that. You at least have a start. So if I open a channel that lets both sodium and potassium move, who's gonna push the other one out of the way and go first? Sodium muscles the potassium out of the way. So more sodium comes in than potassium goes out, and that makes the inside more positive than the outside because sodium has a positive charge. That creates an electrical current. Electrical currents can be made to do work. Okay so far? So what have I done here at my neuromuscular junction? From the time the acetylcholine got released, what happened? Okay, so it diffuses into those subneural folds or junctional folds. It binds to its receptor. Then what? It opens these are gated channels. They are, we say, chemically gated because the chemical acetylcholine opens them. So when acetylcholine binds, that opens the gates. Then what? What goes in? No, the acetylcholine never goes in. It's the sodium. This is a sodium-potassium channel. And sodium goes in preferentially because it's got a stronger drive, electrochemical drive than the potassium does. Okay, so sodium comes in. Sodium's positively charged. The movement of positively charged ions creates electrical current. So the opening of these channels creates electrical current. Okay? Do y'all remember probably in 4th or 5th grade when you were learning to write, you had to write probably a how to do it paragraph? Do y'all remember doing that? I just always say everything happened in 5th grade. So probably sometime in 5th grade you had to write a story about how to do something. And if you wrote your narrative in the wrong order, then I'm hoping you got points off for that. So I have a point to the story, I promise. So if I were going to ask you to write a narrative on how to get dressed in the morning, would you put your shoes on first or your socks? That's a no-brainer, right? Everybody knows you have to put the socks, except my 2-year-old grandson. Never mind that. So you put your socks on first, then you put on your shoes. Well, same goes here. Can I have sodium flowing before I've released my acetylcholine? Can't do it. So you need to know this story as well as any other narrative that to you at this point is common sense, although I promise you when you were 2 it might not have been. Okay? So learn the things in order. So what have we done so far? Starting with a nerve impulse coming down, bringing voltage down to the synaptic knob. What was the first thing we had to do? Here's number one. What did we have to do first? We had to open those voltage-gated calcium channels. Then what? Then calcium comes in. Then that triggers exocytosis. That releases the acetylcholine. Acetylcholine binds to its receptors. That causes the gates to open. That causes the sodium to flow. That creates an electrical current, which is going to translate into a muscle contraction. Does everybody see that that makes sense that way? So if you don't have it memorized yet, that's fine. Don't panic yet. But by Monday, don't you think you ought to know the steps in order? And generally speaking in physiology, that's like the basic first step. Learn things in order. Know what has to happen first before the next thing can happen.
What is it that's not going right for people who have MS?
Okay, we'll talk about that. All right, so hold that question and we're going to get to that. We need to get through the rest of our story of the synapse, I think. I think we'll skip the learning objectives. All right, so what the next part in your book we did yesterday, and that was the sliding filament things. So we've already gone through the sliding filament. So when we get to this picture, where we have to focus our attention is at how we get from the neuromuscular junction to the filaments to make them slide. So if we're looking at just sort of a basic diagram of this, then what we know so far is that the nerve ending is way up here, right? Here's my little synaptic knob. Here's my muscle fiber at the motor end plate. Where are my filaments that are going to do all this sliding thing that we demonstrated yesterday? Are they touching that membrane? No, they're down here. So thin and thick filaments are overlapping and my sarcomeres way over here. So how do I get the signal from the nerve ending to the muscle fibers? That's the question we're going to answer next. Okay? That has a name, actually. It's called excitation, contraction, coupling. So excitation is the electrical activity that occurs at the nerve ending. And contraction, of course, is what happens when those filaments finally start to slide. So what I have to do is connect them to each other, and that's called coupling. The connecting of two processes to each other is called coupling. So we're going to learn how excitation, contraction, coupling is going to occur. Okay? That's the next big piece of our puzzle. Now, we already know a couple of things. First of all, do we know that we need calcium? We need calcium in the nerve ending to trigger exocytosis. We just covered that. But yesterday we said we needed calcium here to make the filament slide. Do y'all remember we went over that? Okay. So I've got to have some way to release calcium inside my muscle cell, and the story so far is that when I have these sodium ions flowing in across my subneural folds, that creates an electrical current. That's where we left that part of the story. So I have to jump from I have electrical current here at the motor end plate to I have some calcium getting loose in my filaments so they can slide. Does everybody see the jump we're fixing to make? Questions about that? All right. So the electrical current can only do work if I have something that is voltage regulated. Does that make sense to you? I need voltage regulated something for electrical current to do some work. On my nerve endings, I had voltage regulated gates, right? They were calcium gates. They let calcium in that triggered exocytosis. okay? What I'm going to find here and here and here, can y'all see where I'm drawing? As I get away from the motor end plate and think three-dimensionally along the entire length of that muscle fiber's sarcolemma, its outer membrane, I have voltage gates. And those voltage gates are sparked by the voltage created at the motor end plate. Okay? So if we just try to see sort of a big picture idea of that, here's my muscle fiber. Here's my nerve ending. So when I triggered the binding of acetylcholine and the opening of those channels that let sodium flood into the cell, I triggered an electrical current there. And that electrical current at the motor end plate is going to trigger the opening of some channels. And those channels are going to be voltage channels, and they're going to be scattered over the entire membrane. And right now I'm drawing sort of a cross-sectional view. So think three-dimensionally. The entire outside membrane of this cell has these voltage-regulated gates. So as the voltage hits the first of these gates, what do? They open. And when they open, they let sodium flow. And the flow of sodium creates voltage, which does what to voltage gates? Opens them. So when these open, ions flow, and that causes the next gates to open. And ions flow, and that causes the next gates and the next gates. Does everybody see where we're going with this? So imagine it as dropping a pebble into a pond. You get a ripple effect. And the entire membrane, from the middle towards both ends, voltage gates open one after another. Everybody picture that okay? Questions about that? Okay, the opening of the voltage gates in and of itself only creates a new electrical current. So by itself, this is unremarkable. We need something else. And what we need is to see what happens to the inside of the cell once we have these voltage gates opening on the outside. So here sits one of those little channels. Here's another one. And if my voltage is coming from this direction, I have a gap in my membrane. Why do I have that? I have some little tunnels periodically that come off of the sarcolemma. Y'all remember them from yesterday, I'm just sure. Those are the T-tubules. Okay, so far? We're going to zoom out and take a look at the whole picture in just a sec. But let's see what happens at one T-tubule. When you look at one T-tubule, what you find it has is, gee, some more voltage-gated channels. So what does that do to the voltage? When it hits the T-tubule, it's going to keep going beyond that because I have more voltage gate channels beyond it. But what happens at the T-tubule? The voltage goes right on down the T-tubule. Okay, so the job of the T-tubules is to conduct that electrical current to the interior of the cell. Are you with me on that big picture? Because when I started things off at the motor end plate, all I was doing was stimulating the sarcolemma, the wrapper around the outside of the cell. But my sliding filaments are deep on the interior of the cell. So how does the inside know when the outside is stimulated? T-tubules are the answer. They're like electrical wires that carry that impulse to the interior of the cell. So when my T-tubules do their thing, well, something else is going to have to happen. There's no point in having electrical current if there's nothing else involved in it. So what we're going to see is that the T-tubules sit right next to the terminal cisterna of the sarcoplasmic reticulum. Guess what's in there? What did I need? I need some calcium. So in the sarcoplasmic reticulum, I keep the calcium I need for my skeletal muscle cells. Okay? How does it know when it's time to get out? I'm going to have some voltage regulated calcium gates, which when the voltage from the T-tubule hits the terminal cisterna causes those gates to open. What does the calcium do? It diffuses out. This is a very complicated story, isn't it? How are we doing so far? I used to actually assign my students to write it out step by step. It takes about 20 steps if you write one thing on a line, about 20 steps. And I finally quit doing it as extra credit, number one, because I got tired grading them, that's for sure. But number two, because I found students would do that effortlessly. Well, no, it wasn't effortless. It was with great effort. So much effort that they weren't studying for the rest of the test. They were just focused on the extra credit, and that was not helping anybody. So they'd flunk the rest of the test and get a perfect score on the 20-question list of the steps, and that's not very useful to you. So keep this in context with everything else, okay? But we spend a lot of time on this because we're going to see similarly next week that a lot of our nerve responses work the same way as our muscles. Okay, so let's go look at the picture that your book gives you, and this is where you remember what I told you yesterday was to be sure to do the homework for this chapter. What's it going to do? It's going to go through this step by step with animations and interactions where you have to put things where they belong to make sure you understand it. All right, so where are we with our story? We had a nerve impulse come down. We had calcium released. That caused the release of acetylcholine. That bound to its receptors on the gates. That lets sodium in. That causes an electrical potential over the entire sarcolemma. Where does that electrical potential go? It's called an action potential. Down the T-tubule. And associated with the T-tubule, what else do you see in blue? Those are the sarcoplasmic reticula, okay? So the calcium gets out of the sarcoplasmic reticula. The calcium gets into the myosin heads, actually to the troponin, tropomyosin, causing the active sites to be exposed, causing the filaments to slide. As long as the calcium is loose, that muscle will contract. So what do I have to do to make the muscle relax? I better get rid of that calcium. As long as the calcium's there, the muscle's going to contract. So I have to get rid of the calcium. So to understand how we get rid of calcium, we have to go reset everything. What did we have to do at our synaptic knob? We need calcium pumps to pump the calcium back out so it would quit secreting acetylcholine. What did we need in the synapse, the synaptic cleft? We have to get rid of the acetylcholine. Okay, once we get rid of the acetylcholine, guess what those gates are going to do automatically? That's just going to let them close, but that doesn't get rid of the sodium that was in there or reverse the electrical potential. So what I'm going to find is that in addition to sodium channels in my membrane, I'm going to have potassium channels as well. And the potassium is going to reverse the electrical charge that the sodium created. And that reversal of charge is going to go down my T-tubules, and that's going to let my calcium gates here close. And that's going to stop releasing more calcium. Is it going to get rid of the calcium that was already there? How do I get rid of calcium? I let it in through gated channels. How do I get it back out? I need some pumps. So you see all this membrane here, and it's chock full of calcium pumps. Okay, so where were the gates for calcium? Here at the ends, the part we call the terminal cisternae that are right next to the T-tubules. But the entire rest of that sarcoplasmic reticulum is full of pumps. Okay, now I want you to stop and think about that. Which one do you think is a faster process, opening some gates and letting the calcium loose, or grabbing the calcium and pumping it back into storage by active transport? Gates, definitely. If you've ever spilled water, you know it's a lot faster to spill it than it is to mop it up. Okay, so likewise, the pumps take longer. So it may take you a millisecond to release enough calcium to cause contraction, but the fastest muscle cells in the body take at least 10 milliseconds to suck the calcium back into storage. So contraction in those muscle fibers lasts G, 10 milliseconds. Because as long as the calcium's loose, muscle cell is contracting. So we divide our muscle fibers into 3 types. We call them fast, intermediate, and slow. And the fast ones are on the scale of about 10 milliseconds. And the slow ones are about 100 milliseconds. That's how long it takes to pump all the calcium back into storage. And the intermediate ones are about 30 milliseconds. Which one has the most sarcoplasmic reticulum, fast or slow? Which one has the most sarcoplasmic reticulum? Fast. Because the more pumps it has, the faster it can pump the calcium back into storage, and the more membrane it has, the more pumps it can have. So when we look at fast muscle cells, we see they are pretty much chock full of sarcoplasmic reticulum. Make sense to everybody? All right, so what I want you to do in your groups now is see if you can go through the whole thing. You need to get all the way through contraction, and then all the way through relaxation. So 2 columns, 1 for contraction, 1 for relaxation. And the one name I didn't give you because I skipped over it was the enzyme that breaks down the acetylcholine. Let me see if I can find that. There it is, up on the screen. This enzyme is called acetylcholinesterase. So step 1 is nerve impulse arrives at the synaptic knob. I'll pop the picture up there in case that helps. All right, so up on the screen, I know it's hard to read it from the screen, but if you're trying to follow along in your book, this figure has basically all of the membrane parts. Okay, so at the synapse, at the neuromuscular junction, you've got, surely as far as the acetylcholine got released, open the channels, lets the sodium flow in. Okay, what are you going to put in the right-hand column about that? What happens to that acetylcholine? Does it stay there? You have to get rid of it. There's an enzyme. What was it called? Acetylcholinesterase. So in your right column, where you're putting the steps that allow relaxation, acetylcholinesterase breaks down the acetylcholine, removing it from the synapse. Therefore, those little channels are going to close. The acetylcholine is gone, so the channels close, and the voltage ends. That ends the action potential. in terms of what's happening at the sarcolemma, you have sodium and potassium channels. And one thing that we'll explain more fully next week, sodium turns things on, potassium kind of turns them off. Okay, so sodium creates an electrical current that starts the action potential. That excites those T-tubules. And then the potassium in the sarcolemma, those potassium channels let potassium back out, and that ends the action potential. Okay, so sodium to start it, potassium to end it. Okay, so that's the beginning and end of the action potential. That action potential goes down the T-tubules. What happens in the T-tubules? When they get an action potential, what did they connect to? Dark blue. That's the SR. So what happened at the SR? They release calcium, and that calcium triggers contraction, right? And then how do I get rid of it? How do I get relaxation? I have pumps for calcium. So wherever you have gates, you gotta have pumps. Gates let stuff out, pumps pull it back into storage. Okay, so once you get the calcium stored back inside the sarcoplasmic reticulum, you should be done. Have you got to that point yet?
So, if we do relaxation, are we starting from, are we going the opposite way, or are we still going?
You're still going from synapse down.
Okay, so then you, okay, that's my
All right, so what do you have for your steps for relaxation? What was your first relaxing step? Calcium pumps where? In the synaptic knob. Okay, that ends exocytosis, which stops any more acetylcholine from leaving. Okay, so get rid of the calcium signal in the synaptic knob. What did we have to get rid of in the synaptic cleft? The acetylcholine. How did we get rid of that? Acetylcholinesterase breaks it, and in its broken form, it can no longer bind to its receptors. So what do the gates do? They close, and that stops the sodium signal. Okay? Along the membrane, we had to get the potassium swapped for the sodium to end the action potential. At the sarcoplasmic reticulum, what did we have to do? We had to have the calcium pumps pump the calcium back into storage. Does that make sense? So in every spot along the way, you're sending out a chemical signal, and then you're either getting rid of it or you're compensating for it, right? So always think of both parts. How do I start it? How do I end it? Are you with me on that? Questions about that? Are we good? Because now we're going to mess things up. We're not done. I'm not done yet. Okay, so if you're caught up in waiting, you can read the screen. And this is going to be our first little clinical case for today. I'm going to explain myasthenia gravis. By the way, myasthenia gravis is specifically assigned to you. It's assigned in that muscle chapter overview that I told you yesterday to be sure to read, because I'm assigning a muscle you weren't assigned in lab. Okay, and it has to do with this particular case. All right, so we're moving on. Ready or not? We're moving on. This is an autoimmune disease. What does that mean? What does auto mean? Self. Auto means self. So autoimmune means you're immune to yourself. You're producing an immune response to your own otherwise normal tissue. For some reason, the immune system gets a message by mistake. We'll look at the details of how that happens in AMP2 when we study the immune system. For now, it's enough for you to know that in myasthenia gravis, you produce antibodies to your acetylcholine receptors at those neuromuscular junctions. And so antibodies bind to the receptors. That blocks them from working. And because the antibodies bind, that triggers the cell to engulf them, to endocytose those receptors with the antibodies. And in addition to other problems that are associated with it, today we're just focusing on what's that going to do that messes up your muscles. So the first thing I want you to tell me is, does that make the muscles paralyzed and relaxed, or overstimulated and rigid? Which one? It's paralysis either way. One's rigid paralysis, the other's flaccid paralysis. So are the muscles unable to contract, completely flaccid, or are they rigid? Are you in a state of rigid paralysis? Are you stimulating the muscles to contract when they're supposed to relax, or are you blocking the signal that's telling them, hey, it's time to contract, and that signal's not getting through? Which one? You're blocking the signal. The antibody binds to the acetylcholine receptor. That blocks the signal from getting through. Acetylcholine cannot bind. Are you with me on that? So the muscles lose tone. They're weak. They have trouble contracting adequately. One of the first muscles that's affected is the one extra one I assigned. Has anybody already looked at that? Well, boy, are you in luck, because I'm going to teach it to you. So that muscle is called the levator palpebrae superioris. And the first thing we're going to do with it is look at the name. A muscle called a levator is named for its action. What does it do? It elevates, okay? A palpebra is the name of an eyelid. You have upper and lower lids, right? So this particular muscle is the levator palpebrae superioris. So which one is it affecting? That would be the upper lid, which in humans is the one that's movable more than the lower one, right? You know, it's exactly the opposite in lizards. The lower lid covers more of the eye than the upper lid. So their eyes are regulated totally different. The wiring has to be totally different from ours. Never mind that. So this muscle is a very small slip of a muscle that just runs from the back of your eye socket forward through the eyelid and attaches to the margin of the eyelid where your eyelashes are, right through the middle of your eyelid. That's not a very thick structure, right? So how many motor units do you think there are in this muscle? Not many, okay? Not that many. But if you impact its ability to contract, then it starts to droop. What is our term for drooping? Ptosis. Thank you for remembering that. So you get ptosis of the eyelid. They just droop. You can't open your eyes all the way. It looks like, we call it half-lidded. Your eyes are just halfway open, and you can't open them the rest of the way. Now, clearly that impacts your vision. And that's usually the first symptom that drives someone to seek help. I don't know what's going on. I can't see very well. My eyes won't open all the way. So once they're diagnosed, then they start running, blood panels and so forth. And what they're looking for specifically is autoantibodies. What's an autoantibody? It's an antibody to yourself, to your own normal tissue. So if they find autoantibodies to acetylcholine receptors, boom, they can diagnose myasthenia gravis. Treatments. There's no cure. Treatments involve blocking the antibodies, production, which that's not very easy to do, removing the antibodies by plasmapheresis. Plasmapheresis is a process by which they take the blood out of your body, they separate the plasma out, they run it through a filter that removes basically all your antibodies because we don't have a way to separate out just the ones that are causing problems, and then they give you all the good stuff back except your antibodies. So since it affects all your antibodies, you have a tendency to get sick from things that ordinarily your immune system would protect you from. But you also have a gland in your neck. It's called the thymus. Have you heard of it? Please don't confuse this with a thyroid. Thymus sits over your heart. It's largest up until about puberty, and then it starts to sort of atrophy, okay? But you may have a tumor of the thymus, or you may just have a hyperactivity of the thymus, and they can do a thymectomy. What would that be? Remove the thymus and hope that the cells that were producing the antibodies abnormally were mostly in your thymus. So if they were and you get it all, then fine, the surgery has helped. It's not a cure, but it's a treatment. But chances are pretty good you're still going to have production of antibodies when you shouldn't. And they may have to do other treatments as well, okay? So why would this be fatal if you didn't treat it? Like a lot of autoimmune diseases, it tends to progress. So you just produce more and more and more. I mean, you're never going to get rid of what's triggering the antibody production. I mean, you need your acetylcholine receptors. So it starts progressing to the point where vital muscles, like the muscles that let you breathe, are finally impacted. And so depending on how quickly it progresses or how good your medical care is, you have swallowing problems, limb weakness, you're in a wheelchair, you're on a ventilator, and then you're gone. Okay? So it is something serious, and it's all based on a mistake that was made at some point, and nobody knows exactly what the trigger is. I have a friend who was just diagnosed with myasthenia gravis. He has his thymectomy scheduled. His dad died of myasthenia gravis. So apparently there's a genetic component to it that I was unaware of until this. Okay, let's skip to the next clinical cases. Here we go. Tetanus. What is it? And here we have a spastic or rigid paralysis. So what's happening here? It's caused by a toxin. These are anaerobic bacteria. What does that mean, to be an anaerobic bacterium? They don't like oxygen. They are what we call obligate anaerobes. If you put oxygen there, it kills them or causes them to fail to thrive anyway. So generally, what do we worry about when we worry about tetanus? What kind of a situation would cause you to think, I need to go get a tetanus shot? A puncture wound of some kind. Okay, not a superficial wound, not a slashing wound, but a deep puncture wound. And the reason is you may be introducing soil bacteria or whatever into the tissue fluid in a way that oxygen can't get to it. Make sense so far? How long can you wait before you decide, gee, I think I'll go get a tetanus shot? Is it an emergency? Do you need to go by ambulance? Now, it's not quite that bad of an emergency, so you can get a tetanus shot a little bit later. I wouldn't wait too long, because if you get a tetanus shot when you've already started producing antibodies, then you get a reaction to the shot, to the vaccine. And so that causes a local inflammatory response around the place where you get the shot. How often should you get a tetanus vaccine? Every 10 years, more or less. Okay, and even at that, wait till you get a puncture wound and then go get your tetanus shot. At least worked for me. I've done it twice, about 10 years apart, amazingly enough. So that worked. I haven't died of tetanus yet, in case you couldn't tell. So what's happening here is a little bit different. The toxin is blocking release of an inhibitory neurotransmitter. And so we'll worry more about that inhibitory neurotransmitter next week when we study how the nervous system controls it. But what I want to ask you is, what's that doing that's causing spastic paralysis? Why is that causing the muscles to contract? So what do you think an inhibitory neurotransmitter does? That's a trick question. It inhibits. It inhibits the stimulation of your muscles. So without that inhibitory neurotransmitter, your muscles tend to contract. Okay? So basically what's happening is you're overstimulating, you're over-secreting acetylcholine. And because your muscles are being overstimulated, the acetylcholine is being released in amounts that you can't deal with. Okay? So am I stimulating or inhibiting the neuromuscular junctions? Definitely stimulating them if I'm causing a contraction. Are you with me on that? Big idea. Okay? Let's see what's next. All right, botulism. Is this something we worry about? Notice it's another Clostridium. What's it doing? How is this different from myasthenia gravis? What did myasthenia gravis do? It bound to the receptor. So if we just kind of draw an overarching picture, if this is the synaptic knob, and here is the muscle membrane, then, and we need some receptors, right? And I need some vesicles. What else do I need? Don't I need some enzyme to break down that acetylcholine? Okay, so in myasthenia gravis, what did we do? We blocked the receptors, right? What are we doing in botulism? Blocking the release. So here, we're stopping the acetylcholine from being released. Okay, so everybody see there are two different things, but they could have an effect, the same end result. You're not getting the signal through. In one case, because you're not sending it, and the other because you're not receiving it. But in either case, it's because you're not getting the signal through. Make sense to everybody? Do we worry about botulism? How do you avoid it? Cook your food. Is that enough? Generally, cases of botulism occur when people eat food that was canned improperly. So it was cooked, it just wasn't cooked well enough. So do any of you home can? Any of you? I do you? So there are some things that are easier to home can than others. Most people will home can tomatoes, no problem, right? Mostly because you grow a lot of tomatoes. Tomatoes are so high in acid that botulism's not usually a concern. But if you ever canned meats or relishes, you never done meat? Just fruit? My husband is a big fan. He's such a domestic guy. Fish, you can can fish. Yeah, well, when you're a fish biologist, you have a lot of fish to eat, so. Okay, so botulism, the organism, again, it's an anaerobic bacteria, so it will go into its sort of hibernation mode by forming spores. And it takes a particular temperature for a particular period of time to kill the spores. So when you get out your little canning guide and it says boil it for 20 minutes, why is it telling you that? Why do you not stop at 15 minutes? You're trying to kill those spores. Can people really die from, do people really die from that? Yes, they do. So the general rule is if you've home canned it and the lid didn't seal, it didn't pop, either throw it out or reprocess it. If you pull it off the shelf and the lid's not sealed, yeah, just don't. If it's slimy, you know not to eat it. But with botulism, you can't always tell. On the other hand, if you have a store-bought can that's bulging at the ends, what do you think? Don't open it and see what it's like. Just throw it in the trash. But in some cases, the botulinum toxin is sufficient to cause respiratory paralysis when you couldn't tell it was bad. You couldn't tell. So you took some home canned green beans and you cooked them up for the family and the whole family died. That has happened. Parents and kids found dead. So,
That happened?
yeah, that really happened. So it's not something I would take lightly. Okay? On the other hand, this is the same stuff that we can charge big bucks for and call it Botox treatments. What's it doing? It paralyzes the muscles. It prevents the release of the synaptic vesicles. Now, Josh pointed out that they're using Botox for migraine treatments. So when I read up on the links he sent, I found out that they don't really know how that works. They don't know. So they're putting Botox injections into the superficial muscles of the head. It's like the back of the head and the sides of the heads. But where's a migraine? Is it on the outside of your skull or the inside of your skull? Migraines? Inside. So how is injecting the muscles on the outside of the skull going to impact a headache that's generated on the inside of the skull? Yeah, they don't really know how it works. But it seems to help some people. It only helps certain people. It doesn't help all migraines. So, anyway, so that was the deal with that. How does it help diminish wrinkles? It causes relaxation of some of those little dermal muscles that I told you a little bit about. A little bit of dermal musculature just smooths out the skin. So it makes it look smooth and not wrinkled. That doesn't make the skin tight or younger looking, in my opinion. But that's a different issue. All right, we did relaxation. We got that. We got this down. But I want to ask you, there was something else I was going to ask you about the synapses. What if, and again, I'm not trying to teach you how to do people in, all right? But this actually happened to a friend of mine. Let me just take that off this way. It was actually a friend of my husband's. They were assessing the safety of rotenone. Have you heard of rotenone? Anybody? Rotenone. Have you heard of Seven Dust?
Yes.
What do you do? What do you use Seven Dust for? Kills what? Bed bugs? Fleas. Occasionally we use Seven Dust to kill it, not on cats. Dogs, yes. Probably there are lots of safer options. Seven Dust is an agricultural treatment to get bugs off your agricultural plants. So if you have aphids on your tomatoes, you can dust them with Seven Dust. Okay, rotenone is used as a fish poison. They actually scatter it in the water, and it paralyzes the fish. So my husband uses rotenone because he's a fish biologist. If they need to find out how many of what kind of fish populations are in a particular lake, they'll take a sample, and the way they'll do it is they'll just rope off one cove with a net, and spread rotenone in that cove, and then for about 3 days they're just scooping fish out of the cove. They can't get out of the cove because the net is there, nobody can get into the cove, and the rotenone just makes the fish just belly up. By the third day, they're pretty stinky, so third day pickup is not their favorite time. But all they're doing is assessing a sample of the population. Doesn't that sound like fun to you, especially in the southeast in the summertime? No. But one of the questions they had was, hey, are those fish safe to eat? Because we've used rotenone on them, so are they safe to eat? So one of the biologists there thought, I'd like to know the answer to that. I think I'll feed one to my dog and find out what happens. And his dog died. So don't try this stuff at home. Turns out the dog had an ulcer in its stomach. So it wouldn't kill a dog with a healthy stomach, but the guy didn't know his dog had an ulcer. So when he fed the dog the rotenone-treated fish, then there was enough rotenone still on the fish to actually enter the circulation of the dog. So bottom line is fish breathe through their gills. We don't, and so we're not inhaling rotenone-laden water. And so it has a very different effect on us. Insects, likewise, they breathe through little breathing tubes. So at that small a scale, the rotenone can get in. Rotenone is an acetylcholinesterase inhibitor. So now, in your groups, tell me what that does. An acetylcholinesterase inhibitor. What's it going to do? Okay, the obvious trick answer is it inhibits the acetylcholinesterase. And now tell me what the outcome of that is. This is not a look it up. This is a figure it out. I know, I'm asking a lot of things. It's great work for 2 hours. All right, bottom line, is it going to cause flaccid or spastic paralysis? Spastic? Who thinks spastic? Is it going to make the muscles contract when they're supposed to or not contract when you need them? Excessive contraction, raise your hand. Excessive relaxation. Okay, defend your choices. What does acetylcholinesterase do? It breaks down the acetylcholine. What does the acetylcholine do? It stimulates contraction. So what does acetylcholinesterase do to contraction? It stops contraction, therefore triggers relaxation. Acetylcholinesterase causes relaxation. I just inhibited it. So you contract and stay contracted. Spastic. Yeah. Push your brain quit working. 10 minutes ago? Do you need me to go through it again? Go through it on your own if you need to, but let's just go through the steps. If I inhibit the thing that inhibits acetylcholine, does it make sense that inhibiting an inhibitor is a stimulator?
Yes.
So in effect, I'm overstimulating acetylcholine. And what does acetylcholine do? Causes contraction. So if I overstimulate contractions, then I get spastic.
Right.
Okay? Good? Questions? Now, it has a different effect in insects and probably in fish, but as we'll find out next week, acetylcholine has a different effect in the brain, and it has other effects in the body besides affecting the muscle. Okay? All right, so we are officially done talking about that, how do muscles contract. Doesn't mean we're done. We're going to do one more thing because we have about 15 minutes. We're going to do one more big topic. Tomorrow, we'll go back through anything that is just sort of out there dangling or still nebulous to you or whatever. But I want to go back and get through this one last big piece, and then we'll wrap up tomorrow, answer any questions, and jump into the new material a little bit early. Okay? So now we're talking about, that's not the one I want. Now we're going to talk about where do muscles get the energy they need. Okay? How much ATP do muscles use? A lot. Every time a calcium pump operates, every time a myosin and an actin separate, every time sodium and potassium pumps are working, 1 ATP per turn. Okay? Every time your muscle cells are making more protein, either to replace or to expand on what you have, every amino acid requires an ATP to attach to a protein. Muscles are enormous ATP sinks. They use a lot of ATP. So you better be ready to produce a lot of ATP to keep them going. Because they are so dependent on ATP, muscles are special in that they can elect, sort of, well, it's not like they have a conscious choice, but they have two different settings, one with oxygen and one without. Now, how well does your brain function without oxygen? It's not going to do it. How about your heart? Nope, can't do it. Okay? Most cells of your body are within easy reach of oxygen and have a fairly low metabolic rate. So it's only an issue if you have an organ with a high metabolism and its blood supply is interrupted for some reason. Okay? So I want you to stop and think about that. If I slice a blood vessel doing surgery on the brain of a patient, brain or spinal cord of a patient, how long before neurons start to die? Really quick, a couple of seconds. And if you have interrupted the blood flow to that organ for a couple of seconds, neurons are already dying. Okay? Now, if I stop the blood from circulating, well, no. If I stop oxygenating the blood, but it's still circulating, so if I have a patient who's stopped breathing, but their heart's still beating, they can last for a few minutes. Or if a patient is not getting enough air, either through obstruction of the airway or if they're drowning or whatever, you can last a few minutes. But still, your heart and your brain require pretty much continuous flow of oxygen through the blood, okay? Your liver and your kidneys not far behind. In fact, renal failure, the shutdown of the kidneys, is not an uncommon outcome of someone even if they've been revived from a heart attack. And one of the most famous cases of that, this is your history lesson for today. Didn't look it up before I started, unfortunately, so I can't tell you when. Those dates, yeah. But Dr. Barney Clark was a dentist who received the first artificial heart, the Jarvik heart. This was in Salt Lake City, and it's been, I don't know, decades. So this was a big deal. An artificial heart in a human body. I mean, it would still make news today. We just don't do it very often. We use heart-lung machines, which are external to the body, but this was a portable device that he was supposed to be able to live and walk around with. And it did have an external part that would activate the pumps. This guy lived about 180 days. How long is that? That's about half a year, maybe 6 months. And if he hadn't had the artificial heart, he would have died almost immediately. And a lot of that's just due to the way we do experimental protocols. They would not have allowed him to receive that heart if he wasn't on the verge of death anyway. Why was he dying? He was dying of heart failure. So his heart was failing, and that was the only thing really wrong with the guy. But his heart was failing so badly that he was on the verge of death. What was he waiting for? A transplant. And one was not forthcoming. So they got permission to put the Jarvik heart in him, and it worked. It worked immediately. It worked quite well. And then he was dead 180 days later. What did he die of? Renal failure. Why? How well was his heart working right before they took it out of him? Not very well. So they waited too late. And he already had renal failure to the extent that it was irreversible damage to the kidneys. And they predicted that had they done it sooner before he had the renal damage, then he may still be alive. Now, the main problem we have with artificial hearts has nothing to do with any of that. It has to do more with the formation of blood clots on the artificial surfaces, which is totally unrelated to today's lecture. So why did I even bring this up? What are the muscles good at that your heart is not, and your brain is not, and your kidneys are not, and your liver is not? You can cut off the oxygen supply to the muscles, and they won't just sit there, they'll keep working. So they have a metabolic pathway that the rest of your body does not have. It's kind of a privileged system. So muscles do have a high ATP requirement. They do have a lot of mitochondria, but those mitochondria are reliant on oxygen being supplied. Now, what in the bloodstream carries oxygen? Hemoglobin. And hemoglobin, of course, is what makes blood red. Muscles, because their metabolism can just outrace the blood supply, have their own stockpile of oxygen. They hold it on a molecule called myoglobin, and just as hemoglobin makes blood red, myoglobin makes muscles red. So the amount of myoglobin in the muscle helps determine how red your muscles look. And so we categorize muscles, generally speaking, into two categories. We call them red muscles and white muscle, or red muscle fibers and white muscle fibers. What is that based on? To a large extent, it's based on the myoglobin. But where there's myoglobin, there's lots of oxygen. Where there's lots of oxygen, there's a high reliance on mitochondria, which tells you that red muscle fibers have more mitochondria than white muscle fibers do. And mitochondria, because they have pigment molecules in them as well, are kind of brown in color. All right, so where do you see myoglobin? Specifically in the redder muscle fibers. We also have lots of glycogen stored. What's glycogen? It's stored glucose. So I can take my glycogen stores and break it down to glucose. And that's something else that muscle is one of the few tissues that actually does that. Most of your cells do not stockpile oxygen or glucose. They don't need to. Their metabolic demands are not that high, and the blood supply is continuous and meets their needs. So what's going on in a muscle that would cut down on the blood supply? Do you remember yesterday I said every time a muscle contracts, it makes it bulge? What do you think that does to the blood vessels that are deep inside the muscle? It squeezes them and makes the blood flow intermittent at the very same time that the muscle's doing the most work. So you're going to dilate the blood vessels that go to your muscles and increase the blood flow overall, but there are still going to be periodic episodes where there may be oxygen coming in, it's just not enough to meet the needs of the cell. Are you with me on that big idea? Okay. So we have another thing that is, if not unique, it's certainly closely associated with muscle, and that's creatine. Have you heard of that? Creatine is sometimes used as a supplement by people who think, gee, if I had more creatine, my muscles would be stronger or faster or last longer or something. There have been very few studies that show any improvement. The main reason there are no studies to show improvement is you can buy creatine anywhere. So nobody's going to make any money off of creatine. No pharmaceutical company is going to be able to market and sell it as a unique trademark substance. So there's not big bucks in creatine. There's bucks in creatine because you can buy it, and there are people who will sell it to you, but what are they going to promise you? Yeah, probably more than it's going to deliver, sorry to say. So you can try creatine and see if it helps you, but the only studies I've seen showed limited improvement in quick movements, like if you're a weightlifter, the clean and jerk, just getting it up there, that may have an impact. And most of the studies I've seen have been kidney problems related to excess consumption of creatine supplements. So like anything else, it's not going to be a quick remedy for anything. Okay, creatine gets broken down to a waste product called creatinine. Creatinine is the waste product of creatine. Creatine doesn't last forever. You have to get rid of the stuff, and your kidneys will excrete it for you. All right, so can you name 3 things that we see in muscle tissues that we don't pretty much see in any other tissues? How did we store the oxygen? We had myoglobin. How did we store the glucose? We had glycogen. And then we had creatine, which we're going to find is a way to have a quick storage supply of high-energy phosphates to feed your ATPs, to regenerate your ATPs. Okay, so what are the big 3? Myoglobin, glycogen, creatine. Okay? Now I'm going to tell you your liver will store glycogen for you, and it takes care of the storage for the rest of the body. Okay? You also do find fat stores inside your muscle cells, so they have their own fat reserves as well. All right, so we have 3 kind of time periods of ATP generation. 1 immediate, 1 short-term, 1 long-term. The immediate way is called the phosphagen system. And in the phosphagen system, what I'm doing is I'm taking my creatine, which has a high-energy phosphate, and I'm just donating that high-energy phosphate to ADP, which is going to leave me with a creatine and an ATP. Phosphagen system is just generating, it's just moving the phosphates from the creatine to the ATP, regenerating ATP. It's like instantaneous. And you have enough, it's called immediate because it only lasts maybe a couple of seconds. So people that are taking creatine supplements are trying to increase the amount of creatine phosphate they have in their muscles to expand on this immediate supply. Our short term is anaerobic, and the reason for that is anaerobic gives you some ATP pretty much instantaneously. So in my short term, I'm doing a process called glycolysis. And in glycolysis, I'm going to break down my glucose to pyruvate, and I'm going to get 2 ATPs. Haven't we talked about this? Yeah. So if I need just a quick energy burst, I'm just going to get these 2 ATPs per glucose pretty fast. The problem is I can't keep doing this forever, because that pyruvate gets converted to lactate. And lactate inhibits glycolysis. So I have to get rid of the lactate. So I'm going to dump it. And when I dump it, that's like taking 1 bite out of a cookie and throwing the rest away. There's a lot of energy left in it. But my muscles aren't going to get that energy because I have to dump that lactate in the bloodstream and let my liver or my heart or my brain cells have the lactate. Has everybody got me on that? So short term only. It's not going to last forever. I can take my glycogen and break it down because I'm using up my glucose reserves a lot faster if I'm just taking one bite and then throwing the rest away. I go through my glucose really fast. So some people refer to this as the glycogen to lactate. Metabolism. So I start with my stored glucose in the form of glycogen. I can pretty rapidly break it down to glucose, pretty rapidly break down the glucose to get 2 ATPs. But then what about the lactate? Lost from the muscle. Okay? So I'm not getting a lot of ATP out of that. So my long-term supply is my aerobic cellular respiration. This is the one that uses oxygen, relies on mitochondria, and that's too much information. So for my aerobic metabolism, I am taking my pyruvate and I'm breaking it down as completely as I can to carbon dioxide. So I'm going to get like 38 ATPs instead of just 2. That's almost 20 times as much ATP. But there's a couple of hooks here. I have to have oxygen. I have to have mitochondria in order to do that. And of course, carbon dioxide is a waste product. So I have to be able to get rid of that CO2. But on the plus side, with aerobic metabolism, I can also burn fats. Okay, so you remember I just said that some of your muscle cells will have fat stored in them directly. They can actually burn fats. And they can also draw fat from the bloodstream. So in general, you're going to do aerobic if you're doing moderate exercise, and anaerobic only when you are doing extreme exercise. That make sense? All right, that's the big picture, and I see that we're about out of time. I'm going to go back through this part tomorrow. Your charge for tomorrow, finish that homework so that you've gone through all those animations and interactions that Connect has to offer. And if there's anything from today's lecture that you're still fuzzy on and you want to go back over, you can ask. If you tell me I need you to go over the whole thing, I'm not going to do that. All right, see you tomorrow.